Hello, everyone. My name is Dana Lorber, and I'm a research associate at the Volk Lab at the Weizmann Institute of Science. Thank you for joining me today, and thanks for the organizers for having me. And I would like to share with you today our findings regarding chromatin organization in vivo. As you probably know, the DNA fiber with a diameter of 2.4 nanometers is wrapped around nucleosomes to create disks with a diameter of 8 nanometers, together defined as chromatin. The chromatin fiber is compacted again and again to allow packing of the 2-meter-long DNA fiber into a 10-micron diameter nucleus. We focus on chromatin's organization at the nuclear scale, also known as mesoscale organization, in contrast to the atomic or single molecule level, a difference of at least 4 orders of magnitude. Deviation of chromatin from its common spatial organization was found to associate with various disease states, such as brain and central nervous system dysfunction, viral infection, cardiac disease, and cancer, implying an interaction between the spatial organization of chromatin and cell function. Hence, it is crucial to study chromatin organization in conditions that preserve or replicate its organization in the native tissue in vivo. The prevailing views suggest that at the mesoscale level, chromatin fills the entire volume of the nucleus, like a bowl of spaghetti or ramen. It is also accepted that heterochromatin, the repressed part of the chromatin, are found at the nuclear envelope, and that euchromatin, the active parts, are found at the center of the nucleus. A more recent model, termed the swampland, suggested by the Kramer family, describes the content of the nucleus as a sponge-like porous media comprised of a chromatin matrix that partitions into a core made of heterochromatin and covered with euchromatin. These descriptions originate from experiments done on culture cells or cells in fixed tissues. But the question is, does these views accurately depict these structures in vivo? To study 3D chromatin organization under physiological conditions, we developed a device that allows us to image nuclei and chromatin in their native tissue in vivo. I designed the minimal constraint device to study stationary and contracting muscles and their nuclei in a live intact Drosophila larva. Basically, the larva is placed in a narrow groove attached to 2 glass capillaries and wrapped in hydrogel. During the experiments, the larva is alive and not anesthetized or paralyzed, and the larva is intact with no impairments, with minimal interference to its natural movement. Note that all the images and movies you'll see in this presentation were acquired using this device. These are 2 examples of muscle and nuclei dynamics during muscle contraction. In the upper movie, you can see the muscle cytoskeleton tagged with GFP, and the content of the nucleus is marked with NLS and cherry. On the bottom movie, you can see the muscle Z lines with GFP, and the chromatin is marked with H2B RFP. Using these tools, we can now look at chromatin spatial organization in the native tissue in vivo. Exploring the chromatin organization in a live nucleus shows that chromatin does not fill the entire volume of the nucleus, but rather organizes at its periphery, leaving a chromatin devoid volume at the center of the nucleus. For the analysis of the peripheral organization of chromatin, we used Arrivis Vision 4D software with a custom Python script written with the aid of the Arrivis tech support team. And following our work, this script is now embedded into their analysis toolkit. To quantify chromatin radar distribution, we used a Matryoshka approach. We divided the volume of the nucleus into 10 concentric shells and calculated the mean intensity of H2B RFP in each shell and normalized it by the intensity of the signal found in the cytoplasm. The red line represents the distribution of chromatin in the nuclei, and as you can see, most of the chromatin is found in the outer shells. This organization was found also in epithelial cells and in salivary glands, but is this organization unique just to Drosophila? In order to answer this question, we imaged human white blood cells, freshly isolated neutrophils, and cultured effector T cells, both fully differentiated. The peripheral organization found in the human cells suggests that this organization is not unique to Drosophila, but rather evolutionary conserved and may represent a general phenomenon. So where can we find L-chromatin? Using mint-body GFP to detect the active segments of the chromatin, we found it to overlap with the chromatin found at the periphery of the nucleus. We wanted to understand the physical parameters that govern chromatin mesoscale organization. One hypothesis was that the nucleolus pushes the chromatin towards the nuclear envelope, as may be suggested from images of fixed nuclei. Imaging live nuclei shows that the nucleolus does not occupy the whole chromatin devoid volume and cannot be the source of this organization. Chromatin occupies 31% of the nuclear volume, and the nucleolus occupies 8%. These ratios were preserved over a wide range of nuclear volumes, suggesting a tight regulation over chromatin volume fraction, the ratio between the volume of the chromatin to the volume of the nucleus. We collaborated with Sam Safran and his postdoc student Gaurav Bajpai, who created a physical model that identified the forces that determine chromatin organization and the parameters that affect them. Sam and Gaurav modeled the chromatin as a polymer, represented as a sequence of beads confined in a sphere. The inner surface of the sphere is covered with lamina, a dense mesh of lamin proteins. Some of the beads, like parts of the chromatin, are lamina-associated domains, LADs, that can bind the chromatin to the nuclear envelope. Sam and Gaurav identified the main forces that determine chromatin organization. The first is the extent of LADs binding to the lamina, pulling the chromatin towards the nuclear envelope. The second parameter is chromatin self-association, pulling it together and phase separating it from the nucleoplasm. And the third parameter is chromatin volume fraction. So what will happen to chromatin organization if we perturbate these parameters? Let's first take a look what happens to chromatin if we change the properties of the lamina. Overexpression of lamin C, the homologue of the human lamin AC, causes chromatin to detach from the nuclear envelope and shift towards the center of the nucleus. This shift was correlated with a significant reduction in the level of active chromatin, another indication for the interaction between the spatial organization of chromatin and cell function. In parallel, the simulations showed a similar trend, where decreased binding of LADs to the nuclear envelope, caused chromatin to detach from the envelope and disperse inside the nucleoplasm. Sam and Gaurav also found that chromatin volume fraction is a major regulator of chromatin organization. As you recall, we showed that in muscle nuclei, chromatin volume fraction is 31%, like shown here, and the model suggests that higher values of chromatin volume fraction will cause chromatin to fill the nucleus. So what might change chromatin volume fraction? In these images, you can see the XY and YZ cross-section through a live nucleus, and where the peripheral organization is clearly seen. Using the common fixation methods causes nuclei to shrink. Nuclei height was reduced by 50%, and nuclear volume decreased threefold. The increase in chromatin volume fraction led to a relatively homogeneous distribution of chromatin in the nucleus as predicted by the model. Contrary to the common notion that fixation is a snapshot of the current biological state, the literature indicates adverse effects of fixation on the cell, such as membrane blebbing, changes to nucleic acids, alternation in the appearance of phase separation, and volume reduction, as shown in this example. So what are the implications of such changes? Using fixed nuclei to study chromatin architecture may lead to misinterpretation of chromatin mesoscale organization. As an example, we use the fly line that contains 2 insertions of multiple repeats of LAC-O sequences in the right arm of chromosome 2. Visualization of these LAC-O sequences is done by crossing this line with another line expressing LAC-I GFP under the control of a heat shock promoter. Exposing progeny larvae to a short heat shock induces labeling of the LAC-O sequences with GFP. In this image, you can see 2 XY sections through the 2 LAC-O dots in the live nucleus, and here you can see the same fly line after fixation. Please note this dot in the fixed sample. Is it indeed in the center of the nucleus? Viewing a cross-section that passes through the 2 LAC-O dots simultaneously, perpendicular to the mast cell, may suggest that prior to fixation, this dot was at the periphery of the nucleus and could not have been inside of it at its center. And just one more thing. The new Top Gun movie is out, starring Tom Cruise. And in one of his interviews, he shared his vision for making the movie, which can be an inspiration for all of us.
You just can't create this kind of experience unless you shoot it live.
To conclude, imaging of live nuclei showed that chromatin does not fill the entire volume of the nucleus, but rather organizes at its periphery, leaving a chromatin-devoid volume at the center of the nucleus. And this organization was found both in Drosophila and in human cells. The model suggests that mesoscale chromatin organization is determined by a balance between chromatin self-association, pulling it together, and LADs binding to the nuclear envelope, pulling it outwards, and is highly dependent on chromatin volume fraction, and that fixation may significantly alter chromatin mesoscale organization and may lead to misinterpretation of chromatin organization. I would like to thank Dalila for enabling this journey. I would like to thank Daria for working with me on parts of this project. I would like to thank Sam and his students, Garav, Omar, Dan, and Amit, for our ongoing fruitful partnership. I would like to thank Sam and Ina from the Cell Observatory for helping us push forward our imaging skills and abilities, and Benny and George from the engineering unit for helping me execute my vision. And I would like to thank you for your attention.
You said that you stressed that this might affect the mesoscale organization, but
Yes.
What do you think about the microscale, much more closer scale, if I call it microscale or nanoscale organization? How much will that be affected? Do you have any comment on that?
We didn't do any work on it in person, but there are several, not many, but there are several papers in the literature that indicate changes to nucleic acids, and both they did both Raman spectroscopy and both imaging experiments, showing that fixation may cause, even in the molecular level, changes. Again, I'm quoting the literature, it's not work that I have done, but there are several indications that these effects go to the molecular level.
Okay, thanks. So in this experiments, could you measure, for example, some specific genes and the distance between them? Like, you could see the overall chromosome organization near the nucleus, but could you also mark some specific gene, which typically they would do like in a fish, but fish is a fixed one?
I'm not really.
Okay.
I wish I could have some kind of fish or oligopanes or something for the life, but if all these methods require a fixation, so we don't do them.
Okay.
Thank you.
Okay.
Yeah, that's very fascinating, actually.
Thank you.
So just a curiosity. So assuming that now all the chromatin is just in the periphery, just in the 30% of the volume of the nucleus, then the nucleolus is still inside. And
right.
What is in the remaining huge space left over in the nucleus?
We all know that the nucleolus or the nucleoplasm is filled with various molecules in various orders of scales, stress granules, RNA in different states and different levels of processes, Cajal bodies, and many more. So we think of it as a reservoir for these molecule.
It's pretty big, right? I mean, it's it looks like that the chromatin is the reservoir for all the rest of the thing. So
okay, that's a
so, but I guess that it would be very interesting to check some of the potential targets that you are mentioning, like because also all these speckles and chiral bodies, all these kind of molecules in general, though that, considering that most of them, they were always being checked by after fixation, it would be interesting to see how and where eventually they do locate in your in vivo conditions. So same way in which you are marking, for example, the histons, maybe like in doing some tagged versions of these proteins, it will be very interesting to see if they have a different kind of localization. It would be, really a blast, no?
I agree. And we're working towards this direction now. And hopefully, we can publish in the near future some of our findings for other molecules except for chromatin, how they organizes in the nucleus, in the live nucleus.
Very interesting.
Thank you. Thank you very much.
So I'm very excited about your presentation.
Thank you.
Several questions. One of them, for example, again about nucleolus. It is very well known that at least in human cells, there is a rather persistent presence of heterochromatin around the nucleolus. It cannot be, and it is known for everybody. That's one. You cannot answer, but I just tell it to you. The other, what about the cell cycles? Again, it is very well known that in G1, the location of the chromatin is such. In SG2, it is another one. What have you imaged? Which cell cycle phase?
We imaged cells in interphase. These are differentiated muscle cells, and they don't divide. So we only imaged cells at interphase. Obviously, when cell divides, chromatin will move from the nuclear envelope towards the center and divide. There's no doubt about it. But in differentiated cells, most of the time, cells are in interphase. And I wanted to add another thing to your comment about the nucleolus. We always see a strand of the chromatin going towards the nucleolus. It's like a balloon on a string. We always see it, but we never saw what you described that is found in human cells, like a shell of heterochromatin covered. Again, this is Drosophila, not human cells, but it would be interesting to look at human cells at the organization of the heterochromatin in conditions that preserve their organization in vivo.
Many people have looked at this, beginning with Miron E, who discovered it around nucleolus. Now, my question is about eventual pulsation, transcriptional pulsation of the nucleus. Do you see anything? How long? How long do you record your cells?
A sampling of the nucleus takes about 10 to 15 minutes. So we won't see cycles of transcription. We record a single point in time. We don't follow it over time.
Yeah. So this pulsation has a medium period, maybe 40 minutes, and it goes from the nucleolus, and your thought that nucleolus is pushing the chromatin. It seems interesting to me. Now I want you to speak about fixation.
Sure.
Which fixation did you use? Why you are sure that your fixation is general and can be, can be extrapolated to any fixation? I doubt very much.
Okay, so First of all, we're using in the lab PFA, which is the common fixation method for Drosophila larvae. But when we saw these differences, we went into the literature. And indeed, in the literature, there's not a lot, but there are indications for the effect of the fixation in various cell types, in various methods, methanol, PFA, and other things that indicate these changes. Being, I think the paper about the alternations and the effect of fixation on phase separation was published, I think, 2 months ago. It's not, when we saw it, we were a bit concerned, so we went to the literature, and we are not the only one that reports the effect of fixation on cells.
Yeah, but no, there are very many nuances. For example, if you take
I agree.
formaldehyde fixation, if you add bivalent ions, if you do the things correctly, you can get quite another picture if you just take paraformaldehyde in PBS. So I disagree with this conclusion. Thank you very much.
Okay. I think that it's fixation. If you do fixation, you have to do it carefully and note the possible changes.
Yes, of course. Yes, of course. Thank you.
Thank you.
About your lemon C overexpression.
Sure.
And this drastic moving, the fact that chromatin moves away from the periphery to the center. In your movies, the lemon C was labeled with GFP, right? But the lemon C stayed at the periphery.
Lemon C, yeah.
So I'm wondering what caused the chromatin to detach if the lemon C is actually at the periphery. So it has this effect at a distance, if you know what I mean.
We thought of a few explanations. We didn't try them. For example, there are several proteins in the lamina that binds to the chromatin, that are located within the lamina and are binding to the chromatin. Perhaps we know from overexpression causes the lamina to be thicker. So maybe now these attachment sites are covered or are less accessible, for example. There is also works that, not in Drosophila, I think, I'm not sure, that there is some extent of lamine within the nucleoplasm itself. So if you overexpress, you'll also probably overexpress them also in the nucleoplasm. We couldn't see it. We looked for it very, very hard, but we couldn't find the increased level of lamine in the nucleoplasm. But these are two suggestions we had. We didn't, pursue them. These are hypotheses.
So there is a question from Vasundhara. So what do you think about live cell imaging techniques where you can add DNA binding dyes and can see a lot of chromatin both in the center and the periphery of the nucleus?
A paper published, I think, about a year ago in Nature Biomedical Engineering showed that if the matrix of tissue culture is soft enough, you can get the peripheral organization also in human mesenchymal stem cells. So the conditions of the tissue culture are very have an immense effect on chromatin organization. So it's not just fixation that can have this effect, but also the rigidity of the surface. So if in this paper they show that if the rigidity is soft enough and replicate biological tissues, you get this peripheral organization. So it would be interesting, and they also they showed in one of the figures, and very impressive, that they can change during while the cells are still growing the cells, they can change the rigidity of the surface, and they start with a soft substrate, and they have this peripheral organization, and when the rigidity increases, the peripheral organization is lost, and chromatin fills the volume of the nucleus. So there's a tight regulation, which is already known. It's not new between the rigidity of the surface and chromatin in cell function, but they showed specifically chromatin organization, which wasn't shown before. So I would recommend people to take a look at that also. That was very reassuring for us also, that you can replicate these conditions in tissue culture.